Oh, yeah. So it turns out you could express all these pieces together, and you could couple sort of paradox and metabolism to reduction of an electrode as the output. And so we were able to basically have electrons come from NADPH, go through our synthetic electron transport chain, and then find themselves out at an electrode. Mm-hmm. And what we ended up doing is using that to build a biosensor for endocrine-disrupting chemicals.
Welcome to Entangled Health, where quantum tech, AI, health, biology, and consciousness research overlap. Josh Atkinson is a professor at Princeton in civil and environmental engineering and in bioengineering, trained as a bioengineer and synthetic biologist, and part of a small global community that builds living electronic devices out of microbes. He and his group design microbes that behave almost like circuit components, turning chemicals in water straight into electrical signals that we can read. Today, we talk about what actually happens in Josh's lab right now, how these living sensors work, and then zoom out to his larger vision of smart, responsive infrastructure, from water systems that monitor themselves to new ways of watching the health of our environment in real time. And a quick disclaimer, these are personal conversations, a co-creation between my guest and me, and nothing of this is an official statement from any company or from my employer or anyone else. Yeah, Josh, thanks very much and welcome to the show.
Thank you for having me.
I think the best is just if you start explaining what happened when you discovered the thing you discovered, so what they are, and just walk us through one day in the lab.
Sure. Yeah, I can give you sort of a primer on, my descent into microbial electrochemistry because I didn't start there. So I guess if we go back to the beginning, I guess I went to university at the University of Michigan. I was coming out of high school. I didn't really have a scope for what research was. I was maybe interested in oncology and healthcare, because of family relations to cancer, right? And so I was basically looking for research, and the very first thing lab that I found myself in was Paul Dunlop's lab, and he works on quorum sensing in the bacteria that live in the light organs of deep sea fishes as well as squid. So these are some sort of famous examples of how bacteria communicate with each other and learn how their population, the number of cells that are in a particular location, so they can do collective functions, right? And so that's where I sort of got hooked on microbiology as well as sort of genetic circuitry and regulation and sort of how cells make decisions and go about interrogating their own personal environment. And so I got obsessed with metabolism shortly thereafter because synthetic biology was becoming a popular field at that moment, and that seemed like a way to have societal impact. Was we're going to go and replace petrochemicals with things we can make from CO2 from bacterial metabolism. Went off to do my PhD at Rice University, starting to work solely in synthetic biology. So that sort of was the focus of the PhD program, as well as biophysics. And that's where I started to get entangled with some electrons and starting to think about redox that's happening inside of the cell. And so I went in just wanting to do pure metabolic engineering, but I found myself doing protein engineering instead, thinking about how basically you could engineer proteins to act as regulators to control metabolism in the cell and try to take all the genetic circuitry, so transcription and translation, gene expression, remove that and just do everything post-translationally at sort of the protein level. And so the thing I got hooked on there were these important redox proteins that deliver electrons in metabolic pathways and how these could be used as nodes to try to control metabolism. And so I started thinking about these proteins are called ferredoxins. They're important for photosynthesis. So all of the electrons that, get excited by the photons in the photosystem find their way into one of these ferredoxins before they get dispensed out into, your plant or your cyanobacterial cell. They're also important for a bunch of different oxidative bacterial metabolisms. And so we started just taking these very simple proteins and asking the question, can you turn them into allosteric switches so you can turn them on and off by adding a chemical input? And so we started building some of these. We got some switches to work. And then I went to a conference to start to talk about this work. And I had a poster. This was SEED. This is sort of one of the premier synthetic biology conferences that happened. I had a poster next to a gentleman named Moshe Baruch. And so Moshe was a postdoc scholar in Carolyn Aho Franklin's group at Lawrence Berkeley National Lab. And what they were doing there was taking this redox pathway from this environmental microbe called Shewanella oneidensis, and they were getting it to work inside of E. coli. So what this protein complex does is it sits in the outer membrane or on the surface of the cell, and allows electrons to come from inside the cell and move outside of the cell to minerals. That's what Shewanella likes to breathe off of. But what they were doing was trying to take this protein complex, put it in a cell that doesn't normally do this, and then hook it up to an electrode to see if they could see electrons coming from metabolism from an E. coli cell that normally doesn't do this. And I was standing right next to Moshe through this whole poster session of, oh, hey, I have this electron transfer switch. You have this electron transfer wire that hooks up to an electrode.
Let's do something together.
These are obviously something we should merge together. And so we actually sort of ignored all the people who were coming to our posters, and we're just having like an ongoing conversation most of that poster session. That turned into a longer-term collaboration and sort of what sparked my interest in sort of the microbial electrochemical world. So Shewanella is not the only organism that does this. It's actually incredibly common in the bacterial world to try to basically get rid of electrons by whatever means you can. So oftentimes we think about our terminal electron acceptor, like maybe oxygen or nitrate. These are the very famous ones that E. coli will use, and we understand sort of regulation related to them. But they're also hooking themselves up to minerals. So rocks that they find in their environment. So oftentimes in aquatic environments, you get tons of oxidation of iron as well as manganese, and they have these manganese and iron oxides that are relatively favorable electron acceptors. And so as long as you can deal with the fact that they're insoluble and they're outside the cell, and you can come up with a way to move an electron on the outside, you can use these as your terminal electron acceptor. And so Shewanella does this, and other famous organisms, Geobacter sulfur reducens, but there are many, many more, both in the bacterial and the archaeal world, that are basically finding these, free sinks of electrons and beginning to use them. And sort of I've now gotten, deeply obsessed with all of the different ways that microbes do.
Yeah, and for somebody who is not so familiar with, let's say, neither physics or biology, so how does this look? What you're building? So is it? I recall you mentioned you're working with some biological wires or these things. Yeah. I tried to explain this to my son. I'm not sure if and the question then is, but how do they look? How long are they? What are they doing? Can I buy them somewhere?
Yeah. These are great questions. So. As with anything in the microbial world, there's an incredible diversity in the molecules that cells use to do this. But to give you some notion of sort of ones we work with, as well as some of the most famous ones. So the organism we work, Shewanella oneidensis, it doesn't make long wires that are made out of proteins. What it does is it extends part of its membrane out away from the cell. And then on that membrane are basically, you can think about them as pores. They're sort of circles. And then within that pore, there's one of these small proteins that has a few hemes in it. And so electrons hop across the hemes, and then they just find themselves right on the surface of the cell. And so the way Shewanella tries to reach out to a mineral is it actually blebs its membrane and extends it out. Maybe, if you're familiar with like pseudopodia or something, it looks vaguely similar to that. This long sort of hair-like thread that comes away from the cell. It's entirely made out of lipids. So it's just the cell membrane with those proteins decorated all over it.
And this is millimeter size, or?
That's so it's typically microns in length. So maybe 1 to 2 microns. They're about the same size as the cells.
Okay. So you need to. Okay, good.
They have to devote some portion of their surface to making these things, so they can't get too large, right?
I was just thinking how you make millimeter long things, which you have to fold the membrane somewhere, but you don't.
Exactly, yeah. Okay. So that's one example for how to do it. I would say that's maybe actually relatively rare, these membrane tubule style of building these wires. A more common set of wires are ones that are made entirely out of proteins. And so this is what Geobacter does. And so it secretes basically a polymeric protein. So you can think about this like, chains that are all linked together, and then they have sort of a heme at the interface of that chain. And these can also grow to be several microns in length. But they're only, I think they're tens of nanometers in diameter. So very thin hairs that are coming off the cells that are entirely made out of protein, but can transport electrons across those hairs. And so Geobacter makes upwards of, I think, 5 different cytochrome polymers at the same time on the surface of the cell. So they have different genes that get expressed, and they all make these, similar wires. They're sort of examples of convergent evolution. You get this very similar heme stacking structure, even though they didn't, necessarily evolve from the same common ancestor.
And I just coming back to this poster session. So did you decide to say, oh, let's build, I don't know, let's build a new sensor, or let's build some living electronics or something like that?
That's basically what happened. So I talked to Moshe at the poster session. We chatted for a few hours that night. I went back up into my hotel room and I just sat down looking like, okay, the system that I have makes sulfide. The system that you make requires a quinone. So how do we get the electron to jump between the sulfide molecule into a quinone? And so quinones are these intermembrane molecules that contain electrons. All of the electrons for sort of your electron transport chain in your mitochondria also uses a quinone. And so I started digging through the literature, what enzymes actually oxidize sulfide and reduce quinones. And there's a whole suite of bacteria called sulfide oxidizing bacteria that very commonly use an enzyme called a sulfide quinone reductase to do exactly this purpose. And so I walked back into the meeting the next day and found Moshe in the crowd and was like, hey, Moshe, we just need to express this one enzyme with both of our pathways and this ought to work. So let's schedule a conversation with our two PIs. Like, let's make this collaboration happen. And sort of it went from there. We had a follow-up meeting the next week. I began ordering genes for expressing these SQRs, as we like to call them, in E. coli, just to see if they would function at all. Also decided, Caroline's group was, the only group really doing heterologous expression of this pathway in E. coli. So I was like, I need to go work with these people to learn how to do this, because it's a non-trivial pathway to get expressed inside the cells. So I started writing some funding applications for a graduate fellowship. I was a graduate student at the time. And so the Department of Energy has very nice graduate fellowships for funded labs from the DOE to go work in the national lab systems. And so the national labs are, I think, probably, America's greatest contribution to science as far as their scope and what they do. They think, very big at the scale that you can't think as an individual PI. And so it was a really great opportunity to go and learn how the national labs work and go work with, the best person doing that type of work. Yeah.
And then you became professor. So something needs to have happened because you became professor.
Oh, yeah. So it turns out you could express all these pieces together, and you could couple sort of ferredoxin metabolism to reduction of an electrode as the output. And so we were able to basically have electrons come from NADPH, go through our synthetic electron transport chain, and then find themselves out at an electrode. And what we ended up doing is using that to build a biosensor for endocrine-disrupting chemicals. And so I had built a bunch of these sensor molecules. Sensor ferredoxins that would respond to endocrine-disrupting molecules. So things like bisphenol A, as well as a bunch of breast cancer drugs and other sort of cancer treatments as well. And sort of we use that as sort of our model test system to show that you could put in an engineered protein to actuate electron transfer from the cell to the electrode. And this happens at really fast time scales. And so typically a biosensor that uses a gene circuit takes maybe a half hour at the fastest, usually typically hours to really get your sensor response. And so this, we're able to see our molecules within minutes. So it just takes diffusion into the cell, and then they can generate enough current that we can detect it at the electrode.
Okay.
So it's fast. It's not as fast as maybe like neuronal signaling. That's maybe pushing it a little faster than what we can do, but maybe we'll get there.
I mean, it also depends on the type of signal, because sometimes you just don't want to have the quick responses, and rather, I mean, if I recall somewhere, I guess in the talk I listened to, you mentioned wastewater things. I mean, these things don't change within milliseconds.
Exactly. Yeah. You don't necessarily need the fastest time scale, but faster than hours tends to be useful.
Yeah. And just help me also. The mechanism behind this is then. We quickly touched the topic of quantum electrochemistry, electron density distribution. Can you walk me a bit through what the hypothesis or the published state is, or what we are going, how this works?
So if we look broadly at electron transfer extracellular in bacteria, the primary mechanism by which this occurs is through redox hopping through cytochromes. So what you end up having there is you have multiple heme molecules that are decorated in these proteins. An electron hops from one heme molecule to the next heme molecule, and it just continues to hop up the chain. And so this ends up looking like a thermally activated process. It looks a lot like redox polymers also look the same way. So, as we increase the temperature, the conduction increases. And so we can measure this in sort of our Shewanella that we work with. We can, deposit them onto an electrode in sort of an ordered way. We can begin to vary the temperature, measure the conductance at each of those different temperatures, derive out conductivities from this. This is a lot of what we spend our time doing, is to try to get these material properties measured for these living biofilms and living materials that we're making. With the idea that we can use this to build, more complex things than just a resistor, which is kind of what we currently build.
And I'm obsessed, not obsessed, but this is my private show, so I'm obsessed with quantum effects in biology. Yeah. And I mean, there is a lot of things. I mean, you mentioned photosynthesis. Nobody really understands how this works. I mean, we understand, but we can't re-engineer maybe. And I was just thinking when you said building the bridge between bio and electronics. There are a lot of things, like variables, what have you, but there are also much more interesting things, which is sensing quantum properties.
Yes, yeah.
Have you thought about this? I'm just to give you one thing before you answer. I had Keeper Sharkey, for example, who detected that, or who mentioned in the talk that, for example, one isotope of lithium is active for in the brain and the other not, which is a spin effect, which is a nuclear spin effect.
Yep.
Have you observed, or have you plans to extend?
Yeah, so I haven't myself worked too much on sort of using these to measure quantum effects, right? But some colleagues of mine have. And so I did at my postdoc in Mo El Najjar's lab at the University of Southern California. He's a physicist by training, right? And so he does a lot of solid state physics in his background, but he does it on biology nowadays. And so anyways, they're very interested in this property called chiral-induced spin selectivity. It's a property that's generic to organic molecules in general, as long as sort of they have chiral centers in them. They can bias the spin of the electrons that are flowing through them. And so they've done some nice work looking at these same multi-heme cytochromes that we work on from the perspective of them effectively being spin filters. And so basically you have a bias towards, spin up electrons flowing through these cytochrome wires. And so they've done some really nice measurements on purified proteins. Basically they'll use Hall devices to sort of do these measurements in an AFM. And they'll also they've now done some very nice work in living biofilms, showing that if you expose them to a magnetic field, depending on the orientation of that field, you have increases in the rates of electron transfer coming out of the biofilms themselves onto the electrodes. And so this is sort of indicating that, this might be something that's real from the biology perspective of influencing how efficient these processes occur. Maybe they're not being exposed to the same type of magnetic fields that we're exposing them to in the lab. They're much higher than you would find sort of out in nature. But what you might end up finding is minerals that have sort of some bias towards the spins that they accept, as well as thinking about the directionality of how electrons flow through these wires. If you can basically build up some spin resistances, you can sort of biases towards moving electrons out of the cell versus taking electrons into the cell. So what I didn't mention is this property is bidirectional, and so some cells use it to dispense electrons, other cells use it to take electrons.
Because I before we started, we mentioned the connection to lice. The other thing is Clarisse D'Aiello. I'm not sure if you know of her.
No.
Clarisse D'Aiello, she was at the UCLA, and now she's doing. She's my go-to person for quantum biotopics. In the episode I had with her, she was of course mentioning the bird's eye navigation. So there's
right. Yep, that's a
cytochrome, etc. And I was really wondering because it was the horse for making noise. Do we have a magnetic sense in these things? And regarding the importance of magnetic components compared to the electric components, do you need these, or can you work with? I mean, you work with electric circuits, so no need for magnetic.
They're one and the same, right? Is electromagnetism, right? So they're always sort of
linked on frequency, yes.
So I say that they are a unified subject here, right? But we've become more interested in sort of magnetic fields and how you can use these from a bioengineering perspective. And so there are other folks who have been working in this space quite heavily of looking at basically some of the same type of redox active proteins that bind flavin molecules. The difference being flavins can be doubly reduced, so you can have 2 electron reductions rather than 1 electron reduction. They have basically magnetic field sensitivities that can be used for actuating some phenotypes in different proteins. So some people have used this for looking at basically increasing magnitudes of fluorescence in the presence of magnetic fields. We've been interested in starting to think about them as basically little actuatable domains that you could use to try to actuate arbitrary protein functions. This is sort of a new space that we're moving into that we haven't done much work on, but it's been something we've been thinking about a lot lately.
And what are you thinking about? Just go on.
Yeah, so primarily to govern turnover of enzymes is really what we're thinking, but as a platform rather than any one particular application. So basically, can you use one of these domains just as a magnetic field sensor so that we can, within a bioreactor, maybe turn on or off one of these metabolic pathways just by applying a magnetic field as sort of a simple stimulus. You could think about this like optogenetics, but using magnetogenetics more or less. Yeah.
Is somebody else working on that, or just
there are tons of people who are currently working in this space. Yeah.
Forgive me my ignorance, but
there's been some very nice papers who've kind of come out in the last 3 to 4 years where people have sort of started to look at these proteins and thinking about this. Typically, this domain that I'm talking about, the love domain, has been used as a blue light activatable domain in optogenetics. And you can make a point mutation that makes it no longer really reply to light in the same way that it does, and now it sort of replies to the magnetic fields. So it has this sort of, it's even in the name of the domain, right? It's a light oxygen voltage sensitive domain, and turns out it's also slightly sensitive to magnetic fields, right?
Yeah. And in your work, your actual work, do you need the classical, the quantum description, or can you work with a classical description? Because I'm just thinking building electronics. And then if we say, let's build electronics or circuits with classical, seems to be easier than using quantum. I mean, even chips work with quantum, of course. But
right. Yes, yeah. So a lot of our work happens at sort of a higher level than the individual molecules themselves, and so classical descriptions are sufficient for a lot of what we're trying to measure from sort of like whole cell biofilm populations of cells and transport through those biofilms. As you push into the actual the molecules themselves and the direct interfaces that they have with the electrodes, some quantum descriptions are actually quite useful in trying to describe sort of how those couplings work. But it's not something that we've had to use in sort of our day in, day out of trying to build a biotechnology using some of these features.
Okay. And let me come to this sensing part. You engineer microbes which detect whatever, something, a signal, contaminant or water.
Yeah, typically.
And then you have a electrical signal which we can kind of read in kind of real time or whatever. And is this? I'm going a bit into not woo, but in the area of speculation. Is this something you think makes them aware?
Yeah. So maybe thinking more of like a consciousness type thing.
A mini I'm just thinking because people are starting to talk about self-replication. There was this news about the Spatzelle, how they were called, and then you build artificial cells that replicate themselves. I think you might have read it. And then now comes the other thing and talks about cognitions, or are we generating God or whatever. Just boiling it down to what really works or not.
Right. So the cells are deeply aware from a biochemical perspective of the electrode. So the electrode is serving as, the main thing they are breathing off of. And so the entire rate of their metabolism is dictated by the potential we apply at that electrode. Okay. If we want to talk about awareness from, the responsiveness of molecules, they are certainly aware of that. Is that, consciousness, that's maybe a different philosophical discussion. But, but they are responding to it. And we have very nice videos of basically looking at the voltages they generate at their membrane in response to these voltages we apply at an electrode. And you get these very strong biochemical responses that are happening. They don't persist beyond, like if you shift the electrode away, this signal disappears. It has some decay time that maybe is something that lingers a little bit about their, yes, we know here. The other thing that Shewanella in particular likes to do is it likes to try to find a terminal electron acceptor to breathe off of. And so it has to make decisions, again, from a biochemical network perspective, on where it should physically be. And so these are motile organisms. They can swim around. There's a property called electrotaxis that people have been investigating with these organisms. And basically, can they sense the electric signature from one of these electrodes? Is this a field? Is this a concentration gradient of a redox molecule? That's sort of up in the air mechanism perspective.
Could it be magnetic? No.
Maybe they have, magnetic field sensors.
No. Yeah, okay.
Yeah, if you have an AC current, yeah, sure.
AC current. Okay.
Yeah. But it's my gut says this is likely a concentration gradient of reduced or oxidized molecules that they are traversing up and down that, just like all the other chemotaxis systems typically work. Now, something that's different, and a colleague of mine here at Princeton works on, is electrotaxis in mammalian cells. And so Daniel Cohen is my colleague's name. And so he's been looking at basically the response of human cells to electrical fields. And so he'll look at kidney cells, he looks at white blood cells, all sorts of different types. And there's a protein that's in the membrane of the cells that now has a fun name called galvanin, that basically, in response to an electrical field, it migrates on the cell surface to one pole or the other pole, depending on the charge and the field that's being applied, and then it causes the cell to drive that way. So this is actually how these cells are traversing wounds in the human body. So you get a small electrical field that gets generated when you have a wound, just from sort of buildup of ion concentrations. And then cells, specifically immune cells, can actually traverse fields of that strength and find themselves where they need to be.
Which opens the door to electroceuticals in a very straightforward thing, just for wound caring.
Exactly. Yeah. And so he has some projects that he's thinking about, sort of, electronic bandages. Him and others. I mean, there are other great folks working in that space as well.
Yeah, as you just mentioned, human things. So I was in the lab 25 years ago, so I did my PhD. And during PhD, you are sometimes in the lab at night. You get lost completely in the topic. So you just, I'm not sure, maybe you entangle with your experiment or whatever. I just don't know. And I recall one thing, when you spend too much in the lab, you get strange ideas. And we have been working with single cells from interosseous from rats. So we were taking the feet from rats and then taking the small muscles. I think it's really a micrometer long. And when you look at these, a colleague of mine had a look at it, and they were shimmering like I'm not sure. Don't know the word for it. Schinken. So if you slice muscles and then you look at it, so you see it shimmer.
Yeah.
And then he said, it also physicist, and then he said, wow, maybe the seat of the soul is in the muscle. And this still sits with me in a very deep sense. But so we didn't put this in a paper, but when you said, so these things sense somehow, or the cells sense something, and they have the tendency to move to along a gradient. I mean, it's a very small gradient. It's very vulnerable because if it doesn't go there, it will die, etc. And this, for me, now looking at all the AI topics, so we're jumping scale, and people claim they are intelligent, etc., but they don't have a body. So what I want to go is, do you think, regarding coming back to this proto-consciousness or whatever, and if it's too woo, we can cut short. But the idea, I was just thinking, do you think with these behavior you observe and building electronics, living electronics, we have found a way to say these things work by itself and they are embodied, and by this they might have a consciousness compared to the other guys that just are digital signals and don't have a body? Do you think there's a fundamental difference, or is it? I'm just asking. So
yeah. So there's a sort of a philosophical question of basically, can you be a conscious or an organ or something with a soul if you are in absence of a body, and then if you were to,
if you cannot die, it's bad luck. Yeah. So
yeah. It's a very interesting question, right? So These bacteria, they certainly they can die, right? That's an element of them. They can't, on their own, aside from deciding whether or not they are there or whether or not their metabolism is active, decide much more than that, right? Like they don't have control over the system, right? They're sort of being dictated to by the electrode that they happen to have found themselves on. And so,
like with a single neuron, I mean.
Yeah. So I guess they don't have this is maybe a different philosophical question, but control over their own fate, right? Mm-hmm. Their fate is being driven by sort of the way that we set them up in our experimental systems. And so, maybe if they were able to freely communicate with us, they could tell us, leave that electrode on, please stop changing the voltage.
Please stop shorting.
Yeah, which we do see in their metabolism, but they're relatively robust, so you can have these things fluctuate and they sit around. Yeah.
And if you scale these things,
so scaling is one of the challenges in these systems in general. And so, all of the early work on engineering these electroactive organisms was done at sort of bulk scale. And so you would be, maybe making a modification in a genome. You'd have a whole strain in a reactor. You'd have one electrode. The cells would, maybe they can form a biofilm. They'll come and attach to it on their own. But you're just sort of letting the biology do what biology does, right? And so it's whatever is already pre-encoded in its genome. It's going to respond in that particular way. That's challenging if you want to go and try to build devices out of these things. And so that's sort of where a lot of the synthetic biology work has been focused to date, is how do you construct devices from these? And so you can do this from, just more of a materials engineering perspective. We can build hydrogels where we trap the cells onto the electrodes. That's one way we can
structures
all that work. Yep. Make a porous electrode, load it with cells, put some skin on it, and the cells will stay there. We've been taking a slightly different approach more recently, which is to engineer the biology to have programs that allow us to have it self-assemble where we want it to assemble. And so we can basically use light to guide the formation of biofilms. So we have sort of a lithographic system for shining light and getting the biofilm to grow in the way that we want it to grow. It helps make that manufacturing process slightly simpler because now our cells are sort of predisposed to doing what we want. This is also the backbone of the entire silicon industry and all of our traditional electronics is the ability to do lithography, right? And make our chips smaller and smaller. Right now, we are limited insofar as we have a cell that's approximately 3 microns long by 1 micron wide, and so we can't get any smaller than that. That's a fundamental limitation of these types of things. But then to start to scale, if you want to build devices that you go and deploy in a bunch of different environments, you can do this in parallel and just build a bunch of small devices. That's perfectly feasible with the technology we have off the shelf right now. But something that a lot of folks have been spending their time thinking about in this field is how do you use the biology as a chemical catalyst and the electrode as a source of electrons to make molecules, right? And how can you do this at rates and scales that are actually feasible to compete with traditional chemical synthesis processes? And this is where scale really is a killer for these types of technologies. And we don't have a solution to this problem currently. So the thickness of the biofilm that you would need to reach the rates that are competitive with a lot of chemical processes, depending on obviously whatever chemistry you're precisely trying to do, is one of the limiting factors for making this actually a tractable technology that can be economically feasible. Currently, it's not there. And so one way is porous electrodes. That's one strategy people have been going after here. Other ways are trying to enhance biofilm formation to make them thicker. And if you can pump up the charge transport pathways that you have from the electrode into the cell, use multiple in parallel. One very useful way of building this electrical to biochemical interface is to just use an electrolyzer to make a molecule like hydrogen or formate, which there are very good electrolyzers these days that make these molecules, and then scale up in just sort of traditional stirred batch bioreactors that we've been using for several decades.
And the biofilms you mentioned. Are they forming themselves? I mean, biofilms, once you have a biofilm, it's quite tough to get rid of it. So are they forming naturally, or would you have to build them, or?
Yeah, no. So that's a good question. So Shewanella will naturally form biofilms. They're relatively weak biofilms, so they're actually easier to get off than some biofilms, like a Pseudomonas aeruginosa is a very strong biofilm. They have tons of extracellular matrix that keeps them adhered. Shewanella uses one large protein to do this and some polysaccharides and DNA on the outside of the cell. On their own, they don't form the most robust biofilms, and they just form them wherever the cells land and begin to divide and begin to populate. And so what we've been doing is basically putting in a genetic circuit to express particular extracellular matrix proteins, and then using light to control the expression of those proteins, which then causes the cells to self-generate a biofilm, but it's using proteins they don't normally use to make biofilms. And so they make a slightly more robust biofilm than the native Shewanella one, albeit it's still weaker than some of your natural multicellular biofilm or multi-species biofilms you find in nature.
Do I understand this? If you want to build an electric circuit or whatever, you would then do also like they do in classical chip production. So you say, I have my whatever surface, and then I put a mask on it, and then I activate it via light, and then there where the shadow is, there is nothing happening. Where the light is coming, they express this biofilm, so I can build really circuit. Are you doing it?
Precisely. That's exactly how we're doing it. We're using the same sort of photomask. Our stuff looks a little bit more simplistic than, say,
not 3 nanometers, I guess.
Yeah. No, I mean, we go to the nanofab to make sort of some of the electrode arrays that we use to do these measurements, and so we use those same types of lithography processes, and so ours is just layered on top of that.
Are these acting then as one big circuit, or are they showing emergent?
So the way that we interrogate them currently, they act as sort of one big conductor is the way I would think about them. You could kind of think about what we're doing here is we have a semiconducting channel, and we're controlling how much dopant is in there. And so we have this biofilm that in and of itself, without the cytochromes, is not intrinsically conductive. And then we control how many cytochromes those cells have on their surface. And so that's sort of our dopant.
And it's just handshake along the thing.
Yep, exactly. And so electrons just hop from cytochrome to cytochrome, and then from cell to cell through that biofilm. So we think about these as sort of conductive channels. Now, if you step away from this and you start to think about if we just kept our electrode poised, how that population of cells is going to be channeling electrons to that electrode, that's where you could probably see some emergent properties that are starting to emerge from sort of their collective needs as a metabolic entity. Yeah, we haven't ever left them on an electrode long enough and looked at sort of like, if the membrane voltages are changing in, heterogeneous ways throughout the population.
Yeah. I had so much things that pop up. I had a recent episode with Neil Thies. I'm not sure if you have heard of him. So he's a pathologist. And a few years back, he discovered he was working with fascia. And fascia, so these things. And it's a bit of. It was at the edge of speculative. It's now clinically published, so it's published. And he has something which he called interstitium, which is the space between other cells. And apparently, this is a body-wide network with connective tissue and a fluid system. In principle, just like the thing you built externally, but just internally. I was just thinking if we can learn from each other, or if, for example, those guys say maybe this network is a kind of a second organ or independent of our nerve system and our endocrine system. There's this interstitium for message transport. I'm trying to come to the word coherence. To build a bridge to coherence, not to orchestrate the whole organism so that just you vibe in the more classical terminology, and with your network or the biofilm network, would this be possible to do experiments that, if those guys come with hypothesis, to say we assume X, and they say, okay, let's build a model of what you assume, and we just measure it, because this seems to be behaving the same. So we have, on the one hand, we have just a system with electrical conductance, and you just have the other thing, but you can build it yourself, and it apparently naturally emerged. I just want to make us think. So what would you be reaction on this?
So the interstitial thing that they discovered has electron transport properties. Is that the idea, or ionic transport properties? Okay. Yeah. So the biofilms that we're building, the matrix proteins that we're having, aside from as much as a protein is a conductor, they're not particularly ionic or electronically conductive. The particular molecules we're using, Geobacter, a different organism, like I mentioned before, uses the electronically conductive molecules as sort of their ECM to some degree. And so I would say yes, in that sense, they do sort of have this, extracellular network of molecules that have transport properties and allow them to sort of transport at distances. They're not necessarily using it for communication, but maybe they are. Yeah, so I think there's a good analogy there to be had. What sort of, I guess, questions they were to ask and whether or not those track is sort of maybe down to the physiology of the organism itself that make it sufficiently different. But I mean, people sort of the way that we think about sculpting a biofilm in this case, people have been thinking about this and tried to doing artificial tissues for many decades, right? This is sort of fundamental for that, and people use the same sort of optogenetic control to sort of build larger scale tissues and organs out of mammalian cells. Again, that's more structural scaffoldy than it is communication and coherence, though.
Yeah. If somebody would come like DARPA or the big funding things and say, we have 5 billion, can you design an experiment for us? What would you do to
compare across those two systems? That's a good one. I would want something where basically we can, in a localized population of those cells, stimulate them in a way so then we can have the communication networks basically transfer to cells at a distance. I think distance dependence in these types of things is what I find to be very interesting. And so even ask these from the very simplistic question of how far away can one of these bacteria be from an electrode and still, sense and feel and use it. And so that's a very simple question, but I don't think we have really great answers. And sort of what are the mechanistic limitations used by the different mechanisms cells use? What dictates sort of what the farthest distances can be? The same could go for communication, right? It doesn't have to just be for energy, for metabolism. And I think that's where fidelity of signal transfer becomes an interesting question. Like as we begin to walk away, if you have this network that's penetrating all of these cells, does the cell population over here feel the same thing with the cell population over here? Is there sort of some network topology that changes how individual populations of the cells experience the signal that's coming from a single sender cell? I think those are the sort of questions I find to be interesting. Yeah. Are there dead zones where you can't transport? And then, how do you overcome those dead zones? Because maybe that's,
limits.
Disease or,
And talking about limits, what's the current practical or engineering failure you hit when you or don't you have limits?
We certainly have limits, and I would say probably the practical one is the amount of signal that we generate, right? So I say these organisms are conductive, right? And they are, but the conductivity of a Shewanella biofilm from our measurements is nanosiemens per centimeter, and so it's not that conductive, right? If you put it on a spectrum, maybe it's closer to an insulator than it is a metallic conductor, right? And so we are practically limited basically on the amount of signal that we can measure. We have to build large devices with many channels for these cells to transport electrons across for us to measure these types of conductivities. And, we were talking about scaling. If we want to downscale our device and use fewer and fewer cells to actually do the same amount of sensing, this is sort of one of our key limitations. And so that's something we've run up against, and sort of we've have a device fabrication setup that works reasonably well for our instrumentation that we use to do these measurements. We use, these research grade potentiostats that, could measure, picoamps of current without issue. We are generating more on the order of microamps per current or microamps per current for our devices, so that it's more in a measurement limit where we don't have to worry about Faraday cages and things like that.
I'm thinking back to the patch clamp. And then a lot of signal noise. And I was just thinking, as I mentioned, patch clamp. So the difference again between, I was having these glass vials. So you have a glass vial, and then you make a very small thing to patch them on the muscle, and you put a silver or whatever into Ringer solution or whatever. So, and then the difference between the wires you build and the just salt conductor. What's the difference between these? So the question is, could we build instead of these patch clamp devices with glass and ringer solution, which is super complicated to get the air out. Instead of this, using these biofilms or the nanowires, would this make sense from an engineering perspective?
Yeah. So sorry, you're saying using the protein wires that these cells generate as coupling with sort of patch clamp type experiments. Yeah, I think that is something that's very much a viable device to build to try to make these types of measurements. The challenge there is generating the protein wires at scale to build devices and in a way that's not so sort of unstable over time. And so some folks have been spending a little bit of time on how do you manufacture more of these wires. That just tends to be grow more of the biomass of the one cell. Some people try to put these in E. coli to synthesize them, in an easier manner. There's been great successes in that space. But I certainly think you can have them function as sort of, sensors for voltages that you might experience.
Have you tried this?
We have not tried this, no.
A colleague of mine, she has a company who's working. Another colleague.
Yeah, I was just at a conference, this forum on new materials, and I'm blanking on her name right now. But someone really gave a wonderful talk on using organic electrochemical transistor devices for measuring. I forget what cell type she was using, but as an alternative.
It really could make sense. Just a thought.
Yeah. But those devices are, a little easier to maybe manufacture at scale, I would say.
Yes.
Organic polymers rather than biology. But
and you just mentioned pico, or what have you, milliamps or whatever, microamps?
Microamps,
and the voltage is what before the thing burns?
Oh, so we operate most of biology operates within plus and minus 1 volt, sort of from the chemistry of biology that it natively does. And so we're operating within that regime as well. So it's very
up to minus
low
1
voltage.
So
we typically, what we're measuring is electrons flowing from the cells to the electrode, and we'll have the electrode poised at a positive potential of about 400 millivolts. So relatively little. And then the chemistry of them oxidizing lactate, it's sufficiently favorable to drive electrons from the cell to the electrode. If we're trying to push electrons from an electrode into a biofilm and then back into an electrode, what we need to do is actually keep the potential within sort of a favorable potential near the redox potential of the multi-heme cytochromes themselves. So this is about minus 100 millivolts. Versus
like normal electrophysiology in principle.
Yeah, exactly. You just need to be near to sort of, keep those Fermi levels near the actual electronic potentials of the hemes themselves. Yeah.
Okay. And just thinking 10, 20 years in the future, what have you built in 20 years?
Yeah, no, that's a good question. So actually, coming off your question of potentials that you operate at, right? Like I'm talking about what we do with Shewanella. There are a bunch of diversity of molecules that interact with electrodes that biology is using. They all have different formal redox potentials. And so one thing that we currently are sort of missing at a larger scale is the ability to do, sensors with multiple channels of communication at different potentials. So thinking about this like we think about, fluorescent proteins, we have different spectra of light. Like we can have different excitation emission spectra for each of these different proteins. We have the same ability with redox proteins. We just haven't been employing them sort of as, multi-potential outputs.
And what would you sense then? So in principle, you would say I have 3 different sensors, and the one is for whatever millivolts and the other one is for whatever, or
Yeah. So like I have a 200 millivolt sensor, I have a 400 millivolt sensor, I have a 600 millivolt sensor. Each of these are hooked up to individual, populations of cells so that we can watch whatever they're designed to sense. So the goal would be to sense more things, and the things that we would be sensing is what we would build protein sensors for. And so these could be 5 different organic molecules simultaneously, or maybe we want to measure a magnetic field alongside of some organic molecules if we had that sensor built, right? I think the main point being is basically multiplexing of sensing in devices. You can do this from the potential perspective by having different, potentials of the redox molecules. You can do this through device fabrication by having smaller and smaller electrodes with more populations of cells that are all individually engineered. But we don't currently have the ability to sort of do that. And so that's where I think, this is heading over probably the next decade more than the next 2 decades. But basically trying to build out more of this.
What could go wrong? So, what could go wrong is that people are bored and it doesn't work, or?
Yeah, I guess finding, the use case where this is, most useful for society will help, drive the work in this space for longer. I think right now we have really wonderful tools for measuring organic molecules. This is what, analytical chemistry has brought us, the ability to have insight into chemical molecules. But it is throughput limited. And you have, labs that are built out to do this. They can do this, at so many samples a day. And we are currently in need of more chemical information than we currently have. So we're seeing this a lot sort of in response to these forever chemicals, all of these polyfluoral alkyl substances, right? This is a place where we now have rules in the United States, at least, from an EPA perspective on drinking water quality, but we do not have the capacity of the LCMS needed to measure it to actually work for all of our wastewater treatment facilities.
So the idea would then be to just deploy the kind of live sensors somewhere and then have them collect something, and then you do just magic upload it in the internet. I mean, that's...
Yeah, more or less. So to have a sensor that's deployed in the environment where we're interested in sensing this, maybe this is a drinking water system, maybe this is a river effluent from that treatment plant, and then have that just continuously be monitoring. Maybe you have to come and replace the sensor with some frequency, right? Maybe they'll foul and they'll go bad over a week or a month or a year. But what you end up getting there is continuous information rather than discrete information that comes from sampling campaigns that are very infrequent and also very costly for sort of facilities to actually operate, right? And so that's been one of the regulatory burdens associated with the new PFAS regulations, is simply that there's a cost associated with measuring these molecules, and there's a limited capacity for measurement within the globe. And so how do we address those two concerns?
So it would be in principle ethical to stay, let's just go pandemic prediction.
Yeah.
Then wastewater, and then you just know if you analyze the wastewater that people are whatever sick, and you detect it in the wastewater first.
Yeah. I think this wastewater epidemiology, or wastewater surveillance, as people will call this, right, is probably one of the neatest things that emerged out of the COVID pandemic, was that we now have capacity to do this. But there are ethical concerns about understanding disease spread within populations, especially if they're not well-mixed populations. So you end up having treatment facilities that are, very localized to one core population, and now you have some idea that they have some disease that maybe has a stigma associated with it. And I do think there are sort of ethical and privacy concerns related to that need to be addressed. I don't know what the right answer there is, right? You're going to have some, I don't know, nowadays we'll have some all-seeing AI that's going to be taking in all this information and making, maybe some decisions about what to do. And then it's a fast-changing world.
Yeah, fast-changing, but I think the ethics is really important discussion, because when you talk about ethics, we have an external ethics advisory board on a lot of topics, also on AI. And the question is always, would you do it even without AI, or even without your thing? And then also, is it ethical not to deploy it?
Right. Yes. Yeah. I mean, this where you could potentially have interventions to overcome specifically diseases that, can spread readily from human to human. Like, should we not be addressing these if we have the ability to monitor when they have upticks in the population?
And these days, always we have to think about cyber. If I would be interested in protecting the data these things produce, can you build maybe with these different sensing proteins that you say the one is sensing this kind of voltage, the other one, can you build fault hack-proof, biohack-proof signals? That just assume in the case we live in a not hopefully far, and maybe not ever coming future, but that people are locked down because in the area there is a certain signal based on whatever. And then
so maybe
you just want to hack it.
Yeah. That's a good question. We've not put much effort into trying to build safeguards for basically a lack of signal. Mostly, a lot of our current challenges are just getting the signals to happen at all in response to the analytes that we're interested in, and not other analytes, right? So specificity is like one of the challenges we're addressing. the way that I would say maybe to think about that is having multiple discrete mechanisms by which sensing of, or, we think about organic contaminants. And so I guess the worst biohack here is you think there's a contaminant when there's not a contaminant. And maybe your population then would act in a way that they don't use drinking water or something. And this would be a negative for society, right? I think what I would like our tools to be used for is sort of the initial indication that there's something wrong, followed by the wonderful analytical chemistry tools that we have. This is basically telling, one of your operators of a facility, we have a problem over here. You should go do a sampling campaign right now. And so that you have a secondary mechanism for doing that sensing. And so I think that would overcome that challenge in a lot of ways. And never relying on that information on its own to make operational decisions per se.
I'm just looking at the clock, and I have a tradition, a kind of tradition in my podcast, that I have a quantum computer picking a random number. And I have already run it, because sometimes you don't know how long the queue is. Greetings to IBM at this. Sure. But I did run it. You want a question?
Sure.
So it did.
Coming from outside.
It did really run on an IBM machine, and it worked. It was not too long in queue. And the question it picked was a one coming from Erik Jensen in Copenhagen. So Copenhagen, IBM, I'm not sure what there's a connection, but anyway. And it is called, what is the most beautiful pattern you have seen in nature?
Most beautiful pattern that I've seen in nature. I'm going to answer this with something that I find very interesting that maybe is not the most beautiful pattern in the world, but it's the thing that I'm obsessed with. So I find the beauty in it that way, right? And so we were talking about electron transport limitations in Shewanella. Another organism I didn't talk about that we mentioned, and actually it was discovered in Denmark, so it is apt to come from someone from Copenhagen, are these organisms called cable bacteria. And so they can form these really lovely cage-like structures on their outside of the cell, and they use them to connect thousands of cells together in a line, and then to transport electrons up that line. And so I find beauty in sort of the simplicity of what this organism looks like, and then the function that it's actually performing, right? It's just a wire, but it's nature's most conductive wire that we've discovered to date. And so this is where I think we're going to, be able to use this to address some of our charge transport limitation questions, but also we're just fascinated in how these organisms operate. I don't know, it's maybe not the best picture. On my wall over here, we have some SEM images of these cable bacteria.
Tell me, because then I think
one of these is actually pretty. This one is not maybe our most pretty image, but
and this is so cool, because I think that's just maybe just going to a closing vision or whatever. So what if you could, are you planning to use these bacteria also productive, or how can you produce them?
So right now the challenge is understanding how these organisms tick. Like, we have a very good mechanistic understanding how Shewanella and Geobacter do their business. We don't have as good of a mechanistic understanding of what these organisms are doing. They're not using the cytochromes everyone else is using. They're using some nickel-sulfur polymer that they're generating. You can see it in Raman spectroscopy very well. They also seem to have charge transport that's not acting in the same thermally activated way at all temperatures. And so this is where they might be using a fundamentally different mechanism for electron transport. And that's why they're interesting. And what I was mentioning earlier, the nanosiemens per centimeter conductivity you get in Shewanella, these guys are, Siemens per centimeter to tens of Siemens per centimeter conductivity. So many orders of magnitude more conductive, and we don't understand how they work. And so we've been trying to get an idea. That other picture didn't come up. Trying to get an idea of what is the molecule that they're making, how can we make it, and then use that to make devices. That's sort of our arc for the next 5 or so years, I think, on this work. Basically, can we synthesize these polymers? This is something that biology has learned to, extract trace amounts of nickel from their environment, turn it into a polymer that's a core part of this, skeleton of this organism, more or less. How do we learn from what they're doing there? Maybe this will help us inform how we recover nickel from the environment. Maybe this will help us make more conductive inorganic materials using, enzymatic processes that are, operating at sort of mesophilic temperatures and pressures, and, maybe a little more green chemistry to get some of the molecules we want at the end of the day.
And can we source this? Or how would this work? So do we need to go to the sea and dig it out, or can we produce these things? I'm just not sure.
Yep. So to date, everyone had been going to the sea and basically bringing home some sediment or going to a lake. They also live in fresh waters. Bringing home sediments and cultivating these organisms. Something that we one of our innovations here is basically coming up with artificial matrices to grow them in. So we basically build our own mud, if you will, to build a barrier here. We take sand, we could take silica, we take organic matter, we mix all of these together into a tube, and we can cultivate these organisms on that substrate. And so we can begin to now, cultivate them indefinitely on something that's chemically defined. Now the question again is like, how do you make a lot of this material? And so scaling them is a challenge. We are doing these in sort of small tubes in aquaria, and so that's something we're working on right now. But you can grow them. You don't have to go out into the environment necessarily to grab them. You can kind of keep them in perpetuity as something you passage from sediment to sediment. And so we're beginning the stages of cultivating them like we do other organisms. But not in the clean exenic cultures that we have for E. coli and Shewanella and Geobacter.
Not in the...
There's some work to be done on the biology perspective there, as well as just a better understanding of what is the molecule that we're looking at and what are the enzymes that produce those molecules, because that we have no notion of right now. Other than that, there's this nickel-sulfur polymer.
I sensed a bit of awe when you showed the picture. So you spent your days engineering. But you spend your days engineering and living things, etc. Try control and measure them, and leaving the lab, digging mud or whatever. What doing? Is there still mystery? Or if you...
Oh, absolutely. I tend to actually find field work to be very fun. So I came up as there are two tracks of microbiology. There's Koch's microbiology of doing things on agar plates and, having one organism that causes that disease. And then there's Winogradsky's microbiology, where you go out in the environment, you get that organism, you isolate it, and you study it as close to the isolation as possible. And so most of my career I spent doing Koch-style microbiology, and I've gotten to do more of Winogradsky's recently, and I found that to be very enriching and rewarding. It's hard. Oftentimes we just use omics techniques to deal with these types of samples. We extract DNA, RNA, proteins. We look at what's there. We make observations about them. The act of interrogating communities that are in the wild is something that is becoming an engineering space in the microbiology world. And so there are people who do microbiome engineering in the human gut, and we're using a lot of those same tools to work in sediments and wastewater facilities as well. So how do you basically deploy a genetic circuit that you've engineered into one of these very messy, very environmental systems? And there's some challenges associated with deployment and booting circuits up in these, hosts that we can't grow conventionally. And that's where I'm finding the most interesting new, engineering work to be done, right? Like we have these awesome molecular biology tools. Now, how do we go and use them out in our agricultural fields or in our sediments to try to sculpt the biogeochemistry that's happening in those environments?
So that keeps you awake?
It does. It keeps me awake, and it's different than building electrochemical devices, right? It ends up flexing an ecology muscle that had been long dormant in my brain that's now coming back, which is fun.
Yeah. Cool. If you would have unlimited resources, and what would you do?
That's a good question. So lately, obviously, interrogating the cable bacteria, conductive mechanisms is the number one thing. And then throwing a lot of energy at this question of magnetic field actuation, and then basically building out the design rules to actuate diverse protein functionalities. A lot of people have spent wonderful time building sensors and these engineered proteins for fluorescent reporters, right? And it was really great for biosensing, but we can use this to control the behavior of biology. You see this in sort of optogenetics and the neuroscience systems. I'm interested in extending that beyond into, metabolic pathways that happen at ecology scales. How can you resculpt the entirety of a microbial community? Starting to think of things that look like terraforming, right? I find this to be very interesting. And, maybe these are useful technologies if we ever want to have an off-Earth presence of how do we get the chemistry to look like the chemistry that's compatible with us? The way to do this is with microbiology. And I mean, the resources you would need to do this on a planet are the ones where you would need sort of unlimited resources. I'm happy to do it in an aquarium in one of our artificial sediments, but eventually maybe this would be something someone 50, 100 years is going to use to try to do.
There's a lot of if the usual suspects listen to this,
right.
Cool. So I very much like the discussion. I would just like to also have my guests to have the last word. So take home messages or wishes or whatever you want to tell our audience. Before I forget, where do people find you if they want to?
Yeah. So I'm based in Princeton, New Jersey. So if you're ever in Princeton, drop me a line. I'm happy to chat with people. But also you can find me on the web on the sort of the usual suspects. I'm present on LinkedIn. I'm present on Blue Sky. My Twitter presence is sort of faded as of late. And then, my email, you can find me at ja4509@princeton.edu. If people want to send an email, that's also great.
Put it in. So good, we have that. And now your final wish or take-home message. And
I guess my take-home message is, if you pull up a gram of soil or a gram of sediment, it may not look like anything interesting, but what you have that's happening inside of that is this dense microbial ecology that's entirely driven, like the key organizing principle is the flow of electrons from one molecule to another catalyzed by these cells. And so appreciate that. You can't see this, but there are billions of electrons that are flowing around, and this is really what makes biology, right? Each of your cells are doing it. They're doing one type of chemistry. Microbes do so many more types of chemistry, and so it's good to appreciate the unseen majority of our planet for those who are not microbiologists.
Yeah, dear friends, we are now entangled, and that's where we leave it, inside the question. I don't know what this opened for you today, but I hope something did. If a question is still moving in you, sit with it a little longer before you decide what it means. Stay kind, stay curious. Until next time, and keep exploring the unseen.